Chapter 15. DNA. God is in the details. For almost 100 years after The Origin was published, living things were studied at the level of limbs or whole organisms. Evolutionists knew little or nothing about the cellular or molecular details. But once they were understood, Darwin's theory ran into difficulties. In this chapter, I shall try to explain why. If an organ could not be formed by numerous successive slight modifications, Darwin wrote in The Origin, his theory would absolutely break down. Natural selection must either work by slight modifications or not at all. Jumps or leaps were not allowed. Then, in Darwin's black box, the biochemist Michael Behe made a case for the irreducible complexity of a particular organ, the bacterial flagellum. That met Darwin's condition. An irreducibly complex organ is one that cannot be formed by successive slight modifications. If so, evolution has been falsified by Darwin's own criterion. Later in this chapter, I shall take a closer look at Behe's flagellum argument. What is undeniable is that in the last half-century, our understanding of molecular complexities has greatly increased, and with it the challenges to evolution. Behe has compared the problem to looking down at the ground from 30,000 feet. From that height, he said, the terrain's details are invisible. It's hard to see the difficulties that creeks and ravines presented to American pioneers traveling in covered wagons. In much the same way, evolutionist ideas are comparable to looking down from an airplane and imagining that a trek from one large feature to another wouldn't be difficult. As science probes ever deeper into the molecular details of life, serious evolutionary thought has been forced to descend from 30,000 feet to ground level, and grave obstacles to undirected evolution have become manifest. Ever more powerful microscopes, instead of illuminating Darwin's theory, have pointed to numerous obstacles in its path. The cell. Not surprisingly, 19th-century naturalists conceived of life in an oversimplified way. Consider the cell, first seen in a strip of cork by Robert Hooke in 1685. It resembled monastic cells, hence the name. But its interior was not remotely understood. Darwin knew nothing of what went on inside a cell. At one point in The Origin, he used the phrase merely of cellular tissue, seemingly confident that the cells themselves wouldn't pose any great difficulties. Some have claimed that Darwin did understand cellular complexity, citing a letter in which he quoted G.H. Lewes, the novelist George Eliot's friend. But Lewes, no wiser than any scientist of his time, was a poor source. He said the cell was a structure of exaggerated importance, with mysterious powers, surrounded by a microscopic lump of jelly-like substance called protoplasm, and the protoplasm lacked any trace of organization. At the same time, the German biologist and Darwin admirer Ernst Haeckel believed that the cell was a homogeneous lump of protoplasm. He went to see Darwin in England and promoted his theory. He also met T.H. Huxley, who said that protoplasm is the physical basis of all life. It is the clay of the potter. Today we know that a cell performs many functions, absorbing nutrients, metabolizing them, manufacturing proteins, dividing itself into two cells, operating in a complex cycle, communicating with other cells, and excreting wastes. The cell contains numerous raw ingredients, including water, carbohydrates, ions, and fats. But organelles constructed of proteins do most of the work. By one account, a typical bacterium, a prokaryote with no nucleus, requires more than 4,000 proteins to function. Biologists have addressed the complexity of the cellular world with various metaphors. Some have compared its activity to that of a town full of people, with its own micro-factories and transportation systems. The British ethologist W.H. Thorpe said in the 1960s that the most elementary type of cell constitutes a mechanism unimaginably more complex than any machine yet thought up, let alone constructed, by man. there may be 50 trillion cells in the human body, or double that number. They've never been counted. Cells, except for cancer cells, also know when to replicate. Within the human body, cells are divided into perhaps 200 types: skin cells, kidney cells, and so on. But others say that 200 is an undercount. Oddly enough, classical genetics began in a monastery with the work of the Augustinian friar Gregor Mendel. In 1866, he proposed the existence of independent factors, thought of as abstract particles, which determine the traits carried by pea plants. The Danish botanist Wilhelm Johansen was the first to call them genes. That was in 1909. He also distinguished between genotype and phenotype. But Mendel's work was not widely noticed until 1900, and Darwin didn't know about it. Later, genes were assumed to be contiguous sequences of DNA and were thought to construct identifiable body parts or traits. But the Harvard geneticist Richard Lewontin criticized that idea, saying we don't have genes for noses. Genes for parts of the body, or for height, size, or traits like intelligence have never been found. The British geneticist Steve Jones said in 2011 that we know of more than 50 different genes associated with height. Most human traits are influenced by so many genes, Jones said, that there is no likely systematic cause and effect. In fact, the gene itself has become ever more difficult to define. Today, genes are defined less in terms of what they are and more in terms of what they do. They help construct proteins. The molecular revolution. With electron microscopes opening up a whole new world, biology departments soon embraced the molecular revolution. After all, a closer look at DNA was far more likely to turn up something new and interesting than one more look at old fossils. In addition, molecular structures haven't changed, at least not since Darwin's time. If the molecular folk need more information, nothing stops them from peering anew into their microscopes. Francis Crick discussed the transition to molecular biology in his memoir, What Mad Pursuit, 1988. The classical period of molecular biology, he wrote, stretched from the discovery of the DNA double helix in 1953 till about 1966, when the genetic code, the dictionary relating the nucleic acid language to the protein language, was finally elucidated. Also in 1966, as it happened, participants in a symposium at the Wistar Institute challenged Darwinism using molecular data. The skeptics argued that it is extremely difficult to assemble a new protein by chance, as Darwinianism requires, because of its huge improbability. Billions of amino acid sequences are possible in constructing a protein, but few are chosen. A wrong sequence means that the protein won't work. The Wistar event was titled Mathematical Challenges to the Neo-Darwinian Interpretation of Evolution. It was something that Darwinism had not encountered before. As we saw in Chapter 13, Bill Gates said that DNA is like a computer program, but far, far more advanced than any software ever created. So what created DNA? Bearing in mind that Darwinism only permits accidental events. Furthermore, natural selection cannot play a role until self-replicating organisms come into existence. Francis Crick, 1916 to 2004. Biologists must constantly keep in mind that what they see was not designed, but rather evolved, Francis Crick said, reminding us of Richard Dawkins. An atheist, Crick signed the Humanist Manifesto and called for humanism to replace religion as a guiding force for humanity. Once rid of Christian beliefs, we could get down to the serious problem of trying to find out what the world is all about. Crick accepted an honorary fellowship from the newly founded Churchill College at Cambridge, but then resigned in protest when it accepted a donation to build a chapel. Like Julian Huxley and Darwin himself, Crick was sympathetic to eugenics. He viewed infanticide as acceptable and argued that rich people should have more children. In 1977, he moved to the Salk Institute in San Diego and took up neuroscience in a forlorn attempt to explain consciousness. In Crick's sequence hypothesis, the nucleotide bases of DNA function like letters in a written language or symbols in a computer code. But as in spelling tests, the letter sequence is important. In the following quotation, Crick discusses the improbability of these events. To generate this miracle of molecular construction, he wrote, all the cell need do is to string together the amino acids which make up the polypeptide chain in the correct order. This is a complicated biochemical process, a molecular assembly line using instructions in the form of a nucleic acid tape, the so-called messenger RNA. Here we need only ask how many possible proteins are there. If a particular amino acid sequence was selected by chance, how rare an event would that be? This is an easy exercise in combinatorials. Suppose the chain is about 200 amino acids long. This is, if anything, rather less than the average length of proteins of all types. Since we have just 20 possibilities at each place, the number of possibilities is 20 multiplied by itself some 200 times. This is conveniently written 20 to the 200th power and is approximately equal to 10 to the 260th power. That is, a 1 followed by 260 zeros. This number is quite beyond our everyday comprehension. Richard Lewontin then pointed out the chicken and egg problem. The proteins of the cell are made from other proteins, and without that protein-forming machinery, nothing can be made. There is an appearance here of infinite regress. What makes the proteins that are necessary to make the protein? That, said David Goodsell, the author of The Machinery of Life, is one of the unanswered riddles of biochemistry. Which came first, proteins or protein synthesis? If proteins are needed to make proteins, how did the whole thing get started? Crick understood that something as complex as a protein evolving by chance was highly improbable. So he became interested in directed panspermia. Once self-replicating bacteria originated somewhere in the universe, they could be spread by intelligent life forms using spaceships to propel the minute critters through interstellar space. Crick used the word directed to imply that someone had deliberately sent bacteria to other planets. His theory was obviously very speculative, he allowed. So he had quietly relocated the problem to another planet. One is tempted to comment. Anything was preferable to bringing God back into the details. Fred Hoyle, 1915 to 2001. The astronomer Fred Hoyle was the same age as Crick and also did most of his work at Cambridge. But temperamentally, they were quite different. In his later years, Hoyle threw up his hands when confronted by the molecular improbabilities and became an open skeptic. He learned something about cladistics from Colin Patterson at London's Natural History Museum and understood the weakness of the case for Darwinism. Hoyle became critical of the belief that life evolved from non-life here on Earth. The odds were too long. Like Crick, he promoted the idea that life on Earth originated in space, spreading through the universe via panspermia. Evolution, he believed, was given a head start on Earth by an influx of viruses arriving accidentally via comets or meteorites. In 1982, Hoyle presented Evolution from Space for the Royal Institution's Omni Lecture. In his calculation, the odds of obtaining the required set of biomolecules for even the simplest living cell without panspermia were even smaller than Crick's. The difference between an intelligent ordering, whether of words, fruit boxes, amino acids, or the Rubik Cube, and merely random shufflings can be fantastically large, Hoyle wrote. He compared it to the probability that a large number of blind men would correctly align a scrambled Rubik's Cube and all come up with the right solution at the same moment. More from Hoyle. So if one proceeds directly and straightforwardly in this matter without being deflected by a fear of incurring the wrath of scientific opinion, one arrives at the conclusion that biomaterials, with their amazing measure of order, must be the outcome of intelligent design. No other possibility I have been able to think of in pondering this issue over quite a long time seems to me to have anything like as high a possibility of being true. Hoyle also published a book called The Intelligent Universe in 1983, so he can be seen as an early proponent of what a decade later became the intelligent design movement. He regarded the notion that the operating program of a living cell could be arrived at by chance in a primordial organic soup here on the Earth as nonsense of a high order. Hoyle re-emphasized the point with his famous comment about a tornado in a junkyard. The chance that higher life forms might have emerged in this way by random events is comparable to the chance that a tornado sweeping through a junkyard might assemble a Boeing 747 from the materials therein. The evolutionary biologist John Maynard Smith responded to Hoyle in The Problems of Biology, 1986. What is wrong with it, he asked. Essentially, it is that no biologist imagines that complex structures arise in a single step. But his reply would have been more convincing if he had pointed to cases where organisms had been observed to assemble themselves bit by bit. That has never been seen. Maynard Smith also said that even proteins formed as a random sequence of amino acids have some slight catalytic activity. But that is now questioned. Just getting the slightest activity requires a protein with stable, functional folds, which is highly improbable. As David Goodsell said in The Machinery of Life, only a small fraction of the possible combinations of amino acids will fold spontaneously into a stable structure. If you make a protein with a random sequence of amino acids, chances are that it will only form a gooey tangle when placed in water. After Fred Hoyle's death in 2001, his assistant, Chandra Wickramasinghe, a Sri Lankan-born astronomer and astrobiologist, said that in the highly polarized polemic between Darwinism and creationism, our position is unique. Although we do not align ourselves with either side, both sides treat us as opponents. Junk DNA. The DNA molecule, with its 3 billion letters, or nucleotides, is conventionally divided into stretches called exons, which carry sequence information for proteins, and, in the higher animals, much longer stretches called introns. These are interposed between the exons, and, when they are spliced out, leave exons in connected segments. The exons are then transcribed into RNA chains that participate in protein synthesis. In 1988, Crick boldly called exons the sense bits, while the excised introns he called the nonsense sequences. The existence of introns came as almost a complete surprise, Crick allowed. Nobody had clearly postulated their existence before experimenters stumbled on them by accident. Classical genetics gave us no hint of them. In What Mad Pursuit, Crick elaborated on the exon-intron distinction. Much of our DNA, perhaps as much as 90%, appeared at first sight to be unnecessary junk. Even if it had some use, its function probably did not depend on the exact details of its sequence. Leslie Orgel and I wrote an article suggesting that much of it was selfish DNA. A better term might be parasitic DNA. The concept of junk DNA strongly appealed to Darwinists, who thought that nonsense sequences were to be expected if DNA was constructed by random mutation. Crick put it just that way. The possible existence of such selfish DNA, or junk, is exactly what might be expected from the theory of natural selection. But this prediction of evolution, like many others, has been dashed by recent research. Human Genome Project. At the start of the Human Genome Project, funded by the National Institutes of Health and promoted by James Watson, among others, the number of human genes was estimated at 100,000 to 150,000. Thus, Scientific American began one story Now that all the 100,000 or so genes that make up the human genome have been deciphered, many of these non-existent genes were even patented. Craig Venter and Francis Collins of the National Institutes of Health then brought everyone down to earth with a very different count, maybe only 30,000 genes. We have only twice as many genes as a fruit fly or a lowly nematode worm, said Eric Lander, a biology professor at MIT and a fellow traveler aboard the Human Genome Project. Today there are thought to be 19,000 human genes, actually fewer than are found in worms. One obvious conclusion is that the concept of a gene is of questionable value. Nature pursued that idea in 2006 in an article titled, "What is a Gene?" The magazine's summary, "The idea of genes as beads on a string is fast fading." The one gene, one protein idea was also coming under assault. We've come to the realization that the genome is full of overlapping transcripts, one researcher said. With the publication of the Encyclopedia of DNA Elements, ENCODE, project in 2012, the idea that most DNA was junk, as Crick had thought, was shown to be wrong. More than 30 papers in various journals, including Nature, Science, and Cell, reported that many of the vast stretches of seeming junk, comprising maybe 95% of the DNA, are the seat of crucial functional activity. ENCODE showed that as much as 80% of the human genome is biochemically active. Junk DNA, once welcomed by Darwinists as proof that evolution proceeds by random meandering, turned out to be another disappointment. Irreducible complexity. Michael Behe made a case for the irreducible complexity of molecular structures in Darwin's Black Box, 1996. He had in mind the bacterial flagellum, a tiny rotary engine attached to certain bacteria and functioning as a propeller, like an outboard motor. The flagellum is composed of numerous parts. Behe's essential point was that every one of these parts must be operational before the flagellum can function. Possessing just some of those parts wouldn't do. Also, the flagellum can't expect the deus ex machina of natural selection to develop the organ bit by bit. One expert who has agreed with Behe is microbiologist James Shapiro. In a review of Behe's book, he wrote, "There are no detailed Darwinian accounts for the evolution of any fundamental biochemical or cellular system, only a variety of wishful speculations. It would seem to be safe to assume that the great majority of molecular machines, not just the flagellum, are also irreducibly complex." Conflict between molecular and anatomical trees. We can construct a tree of life based solely on the basis of similarities of DNA sequences, said Francis Collins in The Language of God, A Scientist Presents Evidence for Belief, 2006. The tree does not utilize anatomic observations of current life forms. Yet, he said, its similarity to conclusions drawn from studies of comparative anatomy is striking. This claim is simply false, says Jonathan Wells. First, DNA-based trees are often in conflict with each other, and they also differ from those derived from comparative anatomy. Here is an example of a conflict between DNA trees. Francis Collins's results showed flying lemurs as closely related to tree shrews, with rabbits and monkeys on more distant branches. Yet a separate study published in 2002 showed flying lemurs as more closely related to monkeys, with tree shrews closely related to rabbits. Jonathan Wells wrote that conflicts based on different DNA-based trees are a major headache for evolutionary biologists, some of whom spend their entire careers attempting to resolve them. Furthermore, phylogenies constructed with DNA often conflict with those based on anatomy. Consider whales. In the 1960s, the evolutionary biologist Lee Van Valen proposed on anatomical grounds that whales are descended from an extinct group of hyena-like animals. But in the 1990s, molecular comparisons suggested that whales are more closely related to hippopotamuses. Discovery Institute's Ann Gauger, PhD in developmental biology, University of Washington, makes the same point. She explained that while baboons and mandrills were once placed together due to morphology, modern phylogenetics based on DNA separates them. Morphology and DNA give different patterns, and groupings based on morphology can be overturned by DNA similarities. There is an additional problem, and to understand it we must return to Darwin's theory, in which natural selection acts on variations. The source of variation was not known, but differences between the generations could readily be observed. Natural selection then went to work, and only the fittest variation survived. That was how evolution unfolded, as Darwin saw it. Then, with the discovery of DNA and its 3 billion nucleotides, the source of variation was more precisely identified. It was mutation. Some of the nucleotides periodically mutated, and mutations were, quite reasonably, construed as errors. They could be compared to typos made by a typist in copying a manuscript, which is then passed on to the next generation. Then more typos are added. Analogously, Darwinians believe that when enough errors or mutations are added, a new species emerges from the old. But that's like believing that if there are enough typos, a poem by Shakespeare will eventually be transformed into one by Wordsworth. David Berlinski made just such a criticism of Darwinism in his book, The Devil's Delusion. Recently, however, we have learned that to preserve the information encoded in the genome, organisms go to great lengths to prevent such errors from appearing. Cells have error-correcting mechanisms built into them. If such proofreading was absent, an organism would soon become little more than a mass of mutations, and not long for this world. At the molecular level, then, we see that what had once been thought of as the source of variation and fitness became instead a source of errors, ultimately leading to speciation. Darwin of the gaps. Kenneth Miller, a committed Darwin defender and professor of biology, said that cells are filled with structures whose detailed evolutionary origins are not known. Therefore, those who argue against evolution might pick nearly any cellular structure, the ribosome for example, and claim correctly that its origin has not been explained in detail by evolution. Such arguments are easy to make, of course, but the nature of scientific progress renders them far from compelling. The lack of a detailed current explanation for a structure, organ, or process does not mean that science will never come up with one. In his 1999 book, Finding Darwin's God, Miller made the familiar God of the gaps argument. Creationists, he said, like to plug God into whatever Darwin didn't explain. But as humans began to find material explanations for ordinary events, he added, the gods broke into retreat. But notice that Miller introduces his own gap argument in discussing the origin of cellular structures. We might call it Darwin of the gaps. We haven't found a material explanation for these molecular details yet, but, he adds in a touching appeal to faith, given scientific progress, we shouldn't assume that science will never come up with one. A work in progress. Molecular biology is still a work in progress, and little that we read today should be regarded as final. Consider the recent and destabilizing claims made on behalf of epigenetics, and then on behalf of horizontal gene transfer. Epigenetics is the study of small chemical markers placed on top of the 4 DNA letters that constitute the genome. Environmental factors help determine where these markers are placed, and they, in turn, influence whether or not a gene is turned on or off. In other words, what is in the environment can influence gene expression. Nature reported in 2013 that in some organisms, these markers are passed on to the next generation. If so, environmental factors affect gene expression not only in the present generation, but also in the next, and maybe the one after that. In 2014, Science magazine writer Jocelyn Kaiser called epigenetics researcher Michael Skinner a heretic for maintaining that chemicals can cause changes in gene expression that persist across generations. But Skinner thought that epigenetics may become the biggest paradigm shift in science in recent history. Some biomedical and epidemiological research has suggested, for example, that a susceptibility to diabetes and heart disease can be passed on by factors picked up environmentally and then passed on to later generations. All this potentially puts us back into Lamarckian territory. We may recall that Darwin resorted to Lamarckism when his original theory was challenged by the Scottish engineer Fleming Jenkin in 1867. Jenkins said that helpful variations occurring accidentally would soon be swamped by unhelpful variations in later generations. Darwin's appeal to a Lamarckian mechanism ran up against the refutation that the offspring of mice whose tails are cut off still have tails. But as we have just seen, epigenetics shows that this refutation of the inheritance of acquired characteristics may no longer be decisive. In lateral or horizontal gene transfer, LGT/HGT, organisms of different species living in the same environment transfer genes from one to the other. LGT, which has attracted attention in science journals recently, is common in bacteria and is thought to explain how they can generate resistance to antibiotics. It's as though immune bacteria are able to come to the rescue of vulnerable ones. According to Alastair Crisp, a researcher at Cambridge University, it appears that horizontal gene transfer has contributed to the evolution of many, perhaps all animals, including humans, giving rise to tens or hundreds of active foreign genes. Crisp went on to say, "The process is ongoing, meaning that we may need to reevaluate how we think about evolution." Casey Luskin essentially agreed with Crisp, adding that gene transfers show that biologists have uncovered genetic data sharply at variance with universal common descent. Common descent implies that genes are inherited from parental generations and not from neighbors. Laterally transferred genes don't fit the standard evolutionary phylogeny, suggesting that the Darwinian tree of life may have to be rethought. Michael Behe said that it's a reasonable deduction to think that some genes were transferred between bacteria by viruses. Viruses seem able to do so by engulfing the cellular debris of bacteria, or by other means. Behe also said that he doesn't think the horizontal transfer explains the origin of a new use for the gene in the new bacterium. One can swap parts between a tool shop that works in the metric system with a tool shop that works with English measurements, but that won't tell you how new complex machinery is put together in the first place. In light of what we know about recent developments, Michael Behe commented that the cell's known complexity has increased immeasurably in recent years, and points ever more insistently to an intelligent designer as its cause. In summary, the tendency of genomics has been to undermine the old Darwinian verities in a way that organismal biology never could. In particular, we have seen the huge improbability of finding the DNA sequences that generate correct protein folding. All the indications are that as scientific progress continues, with more and more complexities likely to appear, the molecular headaches for Darwin's theory will only increase.